Assalamualaikum and hi to everyone. So, let's start with DNA replication definition. DNA replication is the biological process of producing two identical replicas of DNA from one original DNA molecule. DNA is made up of a double helix of two complementary strands. During replication, these strands are separated. Each strand of the original DNA molecule then serves as a template for the production of its counterpart, a process referred to as semi-conservative replication. As a result of semi-conservative replication, the new helix will be composed of an original DNA strand as well as a new synthesized strand. As you can see on the slide, we have 6 enzymes and 1 protein in biology. When you see something end with "ase", it's very possible that it's an enzyme. Enzymes have the ability to speed up reaction and build up or break down the items that they act on. First, helicases the unzipping enzyme. It untwists the double helix at the replication forks, separating the two parental strands and making them available as template strands. Second, single-strand binding protein. Bind to unpaired strands, keeping them from repairing, stabilize the unwound parental strands. Third, topoisomerase. Catalyze in relieving this strand by breaking, swiveling, and rejoining DNA strands. Fourth, primase make RNA primers to get DNA polymerase started. It catalyzes the addition of RNA nucleotides complementary to the DNA template forming RNA primer. Fifth, DNA polymerase III, the replicase enzyme, catalyze the addition of free DNA nucleotides to the 3' end of RNA primer by specific base pairing rule. 6. DNA polymerase I, the repair enzyme, catalyze the replacement of RNA primer with DNA nucleotides. Last, ligase, the gluer. It helps glue DNA fragments together. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and form a replication fork. The separated strand each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase bind to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously. The DNA polymerase adding bases one by one in the 5' to the 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragment. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in 5' to the 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made and the process is repeated again. Once the new DNA has been made, the enzyme isonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up of the fragment of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative. That's it for me. Thank you for watching. Bye.